So, so this is a basic, student microscope. With this light microscope, we already discussed is different components, eyepiece, objective lens, stage, condenser lens, focus, and light sources. So this is a basic components of light microscopy, and each of them is, we have three type of microscopes, like based on the lenses. One is a simple microscope, the compound microscope. So this is like, the stereo microscopes. So the compound microscope have many lenses, like a objective lens. It depends on the magnification. For example, 4x, 10x, 20x, 40x. So this is kind of, based on the what kind of, information you wanted to get from the, images is based on the magnification. If you have a lower magnification, that means the lens, that, you can see the entire area of the samples. Whereas if you use the higher, power, for example, 20x, 50x, or 70x, 100x, then you will get more detailed, informations, but the area of viewing is reduced. So the eyepieces, objective lenses, the stage, the illuminations source, that's important for the components. Eyepiece is so eyepiece is a cylinder which containing two lenses to bring the images to focus for the eye. So the eyepiece we can also call as ocular lens. So this is, you can able to see the images to focus on your eye. So that's the important thing you need to know here is when you look at under the the microscope with the eyepiece, like many people are use. So the magnification you can adjust with the focus. But every time when you observe in different people, the eyesight is different. So the focus and the objective lens, that is the magnification, is same. But when you the person which you look at through the eyepiece, the image will be blurred for one person, and the other person it will be clear, because it is based on their eyesight. So what they can do is they can adjust the ocular lens to their eyesight. So that is the importance of the eyepiece. So the eyepiece is a cylinder which containing two lenses to bring the images to focus for the eye. So the next is objective lens. So as just we discussed, is it projects the magnified image to a fixed position behind the lenses, 4x, 10x, 5x, 40, 20, 80, 100. That's all, how many times you want to magnify, 10 times or 15 times or 100 times. So it depends on the lens which you observe. The next is a stage. Stage is very, very important to hold your samples, the slides. It's a platform, either it's below or above the objective lens, which supports the specimen being viewed. We are going to see the objective lenses is below the stage or above the stage. So two different type of, kind of microscope we commonly used in the laboratory. That we'll discuss later. The important component of light microscopy, another is illumination, that is light source. That is, again, this is also, the below the stage light is provided and controlled in different ways. So this is just for information. Is a basic microscope. It's a single lens compound microscope, that is a more than one objective lenses. Then the stereo microscope. The stereo microscope is also one lens, and it can magnify. So the purpose of the stereo microscope is to visualize the surface of any samples, how it looks, and whether So this is the purpose is just to visualize the surface of the any material. So there are two types of microscope for conveniently they built based on the positions of the light source and the objective lens. So one is called the inverted microscope, the other one is upright microscope. So the inverted microscope means the objective lens pointed upwards so as to view the specimen from below. So that means when you look at this is the stage. So the objective lens is below the stage. The objective lens pointed upwards so as to view the specimens from below. Whereas the upright microscopes are constructed with the tip of the objective points downwards so as to view the specimens from the above. When you look at this is the objective lens, and you can see the samples here. Also, the inverted microscope, the objective lens is underneath the stage, as I mentioned earlier. The light is directed on the specimens from the above. Whereas in upright microscope, the light is directed on the specimen from below. So the upright microscope, based on this, type of microscope, is suitable for the prepared slides. So as you know, when you process samples, transfer onto the slides and glass slides, and then you can observe. So this kind of type of microscope helps for to keep this observe using the prepared slides. Whereas the inverted microscope, the advantage is it's suitable for viewing petri dishes. So sometimes when you grow bacteria, or you can grow cell culture, or you can grow any other fungal or any other bacteria, you can use this type of microscope so that you can able to clearly things. So the advantage is the like a for like a petri dishes, you cannot keep it here because the stage and the objective lens both are very close to each other. So when the petri dish, you can put it this means there is no space. So to overcome this, that's why what they did is the objective lens is put it in underneath the stage. So that is the advantage. The resolution and the quality of image is both are same, but only this is for to our convenient. What type of samples we use, whether we use it for the petri dish that is grow in the media, or here which is prepared slides. That's the basic. As we discussed even last class, the color illumination is important to adjust the condenser lens, the light source, so that you will get brighter images, because it controls the path of light. So that's this. So how we can achieve this is by aligning and focusing the illumination and critically setting the apertures of the microscope to best match the objective lens. So that is the goal of this technique. Also, we have like a bright field microscope. So the bright field microscope, the bright field microscope, which produces an image with the colored specimens having lightened background. Generally, the microorganisms, do not absorb light, but after staining, the organism get the ability to absorb the light. So the bright field is a very common, type of microscope. Any, fungal bacteria, tissue samples you can process, and most of them is colorless. So when you stain with the hematocyanin and eosin, when you use this type of microscope, you can see a colored image. So that is the advantage of the bright field microscope. Whereas the phase contrast, the phase contrast microscope, first described in 1934 by Dutch physicist Zernike, is a contrast enhancing optical technique that can be utilized to produce high contrast images of transparent specimens, such as living cells, usually which grow in cell culture. Also, even you grow microorganisms, thin tissue slices, fibers, latex dispersants, glass fragments, and subcellular particles. So these kind of materials you can able to clearly see through this phase contrast microscope. There is another type of microscope, that is a cone of light. The light source, the specimen must not enter the microscope objective. Then how you can able to see the images is only the light that can scattered by specimens is detected by the objective lens. Also, the everything in a liquid sample, debris, dark field is the best choice to see them. The dark field microscope, the illumination, especially useful for finding cells in suspensions. So the dark field makes it easy to obtain the correct focal plan at low magnification for small, low contrast specimens. So generally, this type of illumination, the initial examination of suspensions of cells, yeast, bacteria, cells, tissue fractions, that all include epithelial cells, chloroplast, mitochondria, even blood cells you can able to see through this technique. So this is not a separate microscope that phase contrast or a dark field, bright field. It's all one microscope, but only thing you have to use the appropriate lenses and objectives. So that is the main components which attach to the regular microscope. So always the purpose of microscope is to magnify your image. Why do you want to magnify your image or any particles or any specimens? You wanted to see the detailed structure that you cannot able to see through your eye. So what we can do is you have to use the microscope so that the different lenses, that is multiple the images, size. So we need to know how many times you magnify the structure of any, your given sample, whether it is a 10x, 20x, 40x, 50x, 100x. How do we know? As observer, you must know that you what kind of image, objective you use. When you take a picture, when you go for software, you can maybe increase the image size because you wanted to see more detailed. So the manipulation is it changes the real, the size of your samples. So for that reason, you need to know the original magnification, the one which you take during the process. So for example, how do we calculate the magnification of your sample? So as we know that we have a multiple objective lenses, that is a 10x, 20x, 40x, 50x, 100x, right? So now always the ocular lens, that is eyepiece lens, that is 10x. So that means when you see through the eyepiece, it shows 10 times higher magnification. Okay. So the next is the objective lens. Which objective lens are you using? Whether it is 10x or 40x or 100x. So the magnification, the calculation is when the image travels through the objective lens and the ocular lens, it magnifies the magnification of the objective lens by 10x. So that means the ocular lens power by objective lens. So that is equal to the total magnification. For example, the ocular lens is always 10 times higher. And then if you use the objective lens is a 4, that means it's a 40x. That means 40 times you are magnifying your. If it is a 10x, if the objective lens is a 100x, that means 1,000 times you magnify your image. So this is how you can magnify. So this is when you take a picture in the software, you can calculate this, what is the magnification, original magnification, that is error bar. So that will gives you when you took the images. So when you observe through the microscope, after you prepare your samples under the microscope, you wanted to see the whole samples. That means if you want to see the whole, means it has to be lower magnification. So for example, you can imagine that when you take a picture with camera or iPhone or any other phone, if you want for long distance, when you look at this 1x, you cannot see detailed, but you can see the entire area with the limit of your phone camera. So the viewing area is more when lower magnification, but the detail is you cannot get it. So if you wanted the target that is far away from you, then you have to zoom it. So the zoom means you have to increase the magnification. So if you magnify, the viewing area is decreased, but the detail you will get more and more when you magnify. At one point, you cannot get clear image when you go beyond the resolving power, because the quality of lens and the resolution is diminished. So that's why the light microscopy, the magnification should be the max is 1,000 to 1,500 or 2,000 times only you can magnify. So always when you go magnification higher, the resolution should be also maintained. Otherwise, you cannot see the image in detail. So always make sure when you take a picture, the magnification, you have to maintain that until you see a clear image. And beyond that, then no use, because the quality of image will be decreased. So make sure that is, when you increase the magnification, what would happen? When you decrease the magnification, what would happen? That's so in general, the light microscopy is, cheaper. You can study both the live and dead cells. You can easy to carry, but nowadays it is a sophisticated microscope is big and you cannot carry. Samples or preparation, you will go for different type of steps, but it is a straightforward. And also you can view the image, the samples you can see a colored image. The disadvantage, it is a good, like the detailed structure, you can get it, and for biopsy lab and other things, diagnostic, this is enough to diagnose the, outcome. But the disadvantage with this is the magnification. As I mentioned earlier, that the magnification, if you go higher after 1,000 times, then you will get a diminished images, quality is reduced. So, because the reason is the resolution. Also, the specimens or surface view of the specimen is also tougher with this. You can, if you use the stereo microscope. So if in that case, how we can overcome all this? So we will study that later. How can you overcome with this? So overall, now we discussed about the basics of microscope. What is light microscope? Why do you want to use a light microscope? And what's the different techniques? So now you have to understand how we can prepare the sample. What is the necessary to prepare the sample? Why do you have to prepare the sample? How you can prepare the sample? The reason for process your sample is observe specimen near natural state as possible. That's most important thing, that the microscope techniques. The reason you wanted to observe the specimen near natural state as possible. Also, your goal is, we really wanted to study any samples, what is going on with the structure or immunological studies. The preservation of many features you should preserve. So your goal is observe a specimen near natural state as possible. Also, preservation of as many features as possible. Also, you should avoid artifacts, and the specimen, when you do the preparation, it should be stable. So this is a basic. So your goal is, when you collect the samples, you wanted to see after the processing your sample, it has to be near natural state as possible. Also, your goal is should be many features has to preserve. So then only you have to get a clear image that the structures. So to achieve this, what you can do? How you can achieve this? One is the preparation, sample preparation. How you can prepare the sample to get better image. So during the process, the sample process, what are all the basic factors which affects when you preserve the samples? Also, very important to note that the buffer plays important role to maintaining the physiological activities during the process. And also what type of buffers we commonly use for the process during the process. Basic factors affecting chemical fixation. pH and buffer. When you process the samples, the stability of the sample all depends on how to preserve, and also the preservatives and other things, the pH is important. Also, how long you preserve the samples, that is also important. So the fixation plays important role to maintaining the integrity of your samples. Our goal is, as we discussed, as many features as possible we have to preserve, and the original, the near natural state as possible. That is most important thing. When you dissect the sample from animal or human, that's biopsy sample. During that biopsy sample, to the process and observe, it has to be maintained, the sample's structure. If you collect the samples, if some reason, if you are not properly processed, then definitely the structure will be changed due to contamination or due to the temperature, due to some various reasons. Then, when you see under the microscope, if it shows a different structure, that means you do not know whether the real structure is that, or due to the improper process, the structure is changed. So always we have to maintain that the integrity of the sample is important. That is how you can achieve is the preservation. So usually we preserve the samples for light microscopy is paraformaldehyde. Also occasionally the investigators, they wanted to use glutaraldehyde along with the paraformaldehyde. So if you process these samples, the fixatives is important and the dilutant is important. So how we can dilute this by concentrated fixatives if we can use buffer. And how do you wash your sample? It requires buffer. Also, like the affecting during the process, the temperature. Temperature always important because some chemicals is stable, some chemicals is unstable. So we have to maintain the temperature. Also the size of the samples. Because, like when you do the biopsy, or if you have a bigger piece of samples to analyze, means you cannot do the entire, tissues or organs to observe, or you can process. So the specimen dimension is important when you take a sections. That's a 3 to 4 mm. Also the preservatives or any other, you have to completely immersed so that it will be, preserved, all of them. Also, how long you wanted to keep to fix the sample, that's important. So minimum, you should incubate this for 2 hours, and then you can decide based on your, availability. Like if you want to do some other time, put them in the cold room, so that you can, come back and do analysis. So these are all very, very basic, important factors that will be, affected during your process. So what is a buffer? A solution containing a weak acid and its salt served to hold pH steady during the fixation process. So that is important, the buffer is a solution which contains a weak acid and its salt that is served to hold the pH steady. So to one point, the proteins have isoelectric point where the number of plus and minus charges are equal. So that is the point. The fixation is more effective. So based on that, when the buffer plays important role because you can dilute the fixatives, wash the samples, so that it will keep the physiological activities become normal. So for that, the buffer we can use. So there are different type of buffers commercially available, and we are not going to see all of them, but a few of them we can show how this will be used for our experiments. So a buffer must be selected which is effective at the pH desired. So that's most important thing, when you take some example, one buffer, for example, PBS. If you take that and, used for, frozen sample, we have to think twice. So we have to check based on the previous report, what pH it require to fix the samples. Second one is compatibility with the fixatives, components, and strains. So the compatibility is always important, to fix your samples. Next is, the glutaraldehyde. Paraformaldehyde is the ideal fixatives. Occasionally, the glutaraldehyde also used. This one always react with the sulfhydryl group and phosphate buffer, which is precipitate uranyl acetate. So commonly employed in TEM with this type of, experiment. We will discuss this, some other day. Also, when you use buffer, it should be effectiveness at a low concentration salt. Most buffers begin to lose their capacity to control pH as they are diluted out. So the most buffers, if you think that, the salt is not good, means if you dilute it too, that is also not good. If it is concentrated, that is also not good. So the most buffers begin to lose their capacity to control pH as they are diluted out. So if you do that, the low concentration means, some buffer, which is cacodylate and, phosphate buffer is not ideal candidate for, the low concentration of salts. So if buffer is to be used to rinse or temporarily hold specimens prior to fixation, it should support life, right? But not should be toxic. So when you biopsy taking from humans or collect the samples from animal or in the laboratory when you grow, tissue culture, your goal is you have to hold the specimens temporarily prior to fixation. It should support life during that process. So if it is good, it support life means how we can use this. So we called it a solution, Ringer solution, for temporarily maintaining the normal physiological conditions of an organism. Also, the cacodylate and the collidine are very toxic. These are all two important, buffer those days. That is very, very toxic. So some important compatible buffers are PBS, TRIS, HAPES, PIPES. So we are going to see some buffer, in detail. So one of them is sodium cacodylate buffer. So this buffer is effective. The pH range is 6.4 to 7.4. So this buffer lacks phosphates that could interfere with the cytochemical studies. This buffer, when you use for electron microscope, it is not compatible with the stain. So this type of buffer we commonly use in the animal tissues. Also, this buffer is contain arsenic, so we are not recommending often to use this type of buffer. Another important buffer is phosphate buffer silane, which is the effective range is 5 to 8. That is a wide range and more physiological than the other buffer. So this type of buffer, buffering system, we found this in living systems in the form of inorganic phosphates and esters. So the phosphate buffer, PBS, is non-toxic to the cells when you grow in culture. It's in vitro studies. Not as sensitive to temperature changes. When you wash this buffer with any samples, it holds the physiological activities, and also it is not sensitive for heat. Even if you go for 3750, still the buffer is active. The buffer cause artifacts such as electron-dense particles or nuclear shrinkage, so that it's tough when you use for electron microscope. So some buffers, we infrequently used is veronal acetate. The effective buffering system is between 4.2 and 5.2. Also should not be used with aldehydes. Also, the veronal acetate does not precipitate with uranyl acetate. Preserves this buffer well when you used osmium tetroxide. So the osmium tetroxide is one of the fixatives. Then we will discuss that when we discuss in the electron microscope sections. Also collidine, that is another buffer, which is also effective around the neutral pH. And again, we use this for formaldehyde. Also, it does not react with the osmium tetroxide. So we cannot recommend this for electron microscope. There is an important buffer is also the TRIS. It's a poor buffering capacity when it is acidic, and it's biological inhibitor. This also reacts with glutaraldehyde and causes excessive extraction of cellular components. So that for these reasons, we cannot use this for or recommended for electron microscope studies. There is another buffering system is a heaps buffer, which is effective at neutral pH. The temperature is 37 degrees centigrade, and it may interfere with amine aldehyde reactions. As we mentioned earlier, that the fixatives for light microscopy is paraformaldehyde. So the paraformaldehyde is available either powder or aqueous solutions. The aldehyde is, low molecular weights. That's why it is one of the best penetrating fixatives all of the other fixatives. Also, this paraformaldehyde is widely used in fixation of resistant materials such as seeds, spores, plant materials, etc. There is another fixative called glutaraldehyde. The glutaraldehyde is commonly used for electron microscopy, but occasionally the investigators also used along with the paraformaldehyde. Because it is widely applied fixative in both the TM and SEM and highly cross-linking of all the aldehydes. It providing the best ultrastructural preservation in the widest variety of tissue types of any known chemical fixatives. So far the glutaraldehyde is one of the best fixatives all time for electron microscope. So which fix the nucleic acids, this will fix many of the features, but not nucleic acids directly, especially. But may be retained due to the preservation of associated proteins in eukaryotes. So that is the advantage of the fixatives is, the glutaraldehyde, it's not directly fix the nucleic acids, but associated proteins, once it fixes, automatically that will go fix the nucleic acid related proteins. So that is the, important features of the glutaraldehyde. Also the soluble carbohydrates also not preserved, but some glycogen may be retained. So it is advisable to use distilled glutaraldehyde prepared specifically for electron microscope. So what is the action? Anyway, we always talk about preservation. What is the preservation? When you look at this, when you go for grocery supermarkets, you can able to see three different type of storages. One is room temperature. Then next is refrigeration, that is a 4 degree. Then third one is -20. So why do you want to have three different type of temperature? One is -20, the other one is 4 degree, the other one is a room temperature. So the reason is some fresh product that will stay for 1 day or 2 days at room temperature, then will not be a problem. But if extended the days, then there are some microorganisms, presence on the surface of this material, things that will start, decaying these, contaminate the product, and it's spoiled. So to avoid this, if you put this product into 4 degree, that will control the microorganisms production, because it's a 4 degree, it won't grow. So for some 1 week or 10 days, you can keep temporarily fix this. The third one is -20. You can stay for 2-3 weeks if you keep your samples at -20. It won't spoil. So that is the strategy here also. So we talk about the preservation means we avoid the contaminations. So that means how then it fixed the glutaraldehyde or paraformaldehyde or osmium tetroxide. So here the glutaraldehyde, which reacts with the lysine, tyrosine, tryptophan, phenylalanine, histidine, cysteine, proline, serine, glycine, arginine. So these are all the amino acids. It's directly interact with this and, stop further act on this. So it is important, particularly to match the osmolarity of your tissue to avoid the distortion. That is another important thing. So the preservation means when you add the chemicals that will penetrate into your tissues and fix the different amino acids, or to arrest the different amino acids, go further activities. Again, the another same type of like preservatives is osmium tetroxide. So it's also commercially available, which is non-coagulative type of fixatives, but it lacks the ability to cross-link many proteins. Very poor rate of penetration. This is because lack of ability to cross-link. So its use of as a primary fixative is quite limited because of that reason. So although it is popular in some mixtures with other fixatives for unicellular organisms. So we will discuss about this, how important the osmium tetroxide when processed for electron microscope. So how does this work? How do you, this osmium tetroxide fix is it reacts with primarily double bonds and sulfhydryl groups of different type of proteins, causing major conformational changes in the structure of proteins. The osmium tetroxide also causes tissue hardening. That's advantage, like when you fix with the osmium tetroxide, it not only preserve the samples, but also which gives a strength to the samples. It gives a hardening, so which impacting mechanical strength and affording some extra resistance to shrinkage and distortion during subsequent steps. So that is important as a point to note that here the osmium tetroxide not only to fix the samples but also harden the samples. That gives you, when you take a sections, it gives you the mechanical support. So that's the advantage of the osmium tetroxide. So now the first step is fixation. So overall, the process for light microscopy is fixation, dehydration, infiltration, embedding, sectioning, staining, and observation. So totally 7 steps is involved when you process your sample. So now, first we discussed is fixation. So one more time, I'm repeating the protocol, the process. Your sample for light microscopy is 6 steps: fixation, dehydration, infiltration, embedding, sectioning, staining, observation is the 7th one. So keep this in mind. It is very important. So once you fix your sample, what is the next step is dehydration. So the dehydration is very, very important and serious if we dehydrate. So every time when we go to primary care physician or in emergency, immediately they check our lips, eyeball movement, and um, things. Do you know why they do all this? They wanted to check whether you have dehydrated. So what would happen if you dehydrate? That's most important thing. So especially when people are sick, or if they are not drinking enough water, or during summer, or when they go for beach, or um, when you lose the water content, what would happen? So as you all know that the blood is, most of them is water content. The blood is carrying oxygen, right? The hemoglobin goes to all the cells to provide the oxygen. If the water content is slowly evaporated, or goes some dehydrated, means water content is slowed down, then the blood flow will be affected. If the blood flow is not go through all the tissues, cells, then we started the cells are dying because of there is lack of oxygen. So that's why it is very, very important. We should not dehydrate. So that's why immediately they put an IV and things to put them saline. That is a phosphate buffer saline, PBS. So, but whereas here now we know the importance of the dehydration. If you remove the water content, you will affect. But whereas here, we intentionally remove the water content from the samples. You may ask, what is the purpose you wanted to remove the water content from the cells? As just I mentioned, what would happen if water is removed? Then definitely that will also affect the structure of the samples and quality of your samples. So what is the necessity then you need to dehydrate? So the dehydration is, if the sample has water content in the cells, when you go for further processing, that will interfere with the subsequent steps, because we use many solvents and reagents and other chemicals, so that you cannot able to properly process the samples. So to avoid this kind of things, we should remove the water content without affecting the samples. So as we know, the cells are filled with water and other things. If you remove the water content immediately or suddenly, then the cells will be shocked and it will be shrink. Also, when you process samples without dehydration, when you go and observe under the microscope, because you use a light source, it will generate heat. So once it penetrates the tissues, because if there is a water content, it will be evaporate. If it evaporates suddenly, it will shrink the sample structure. So that that is not our goal. We wanted to maintain the integrity of the samples as natural as possible, and also as many features as possible we have to fix it. So our purpose is here, we are going to intentionally remove the water content without affecting the structure of the cells. So then how we can achieve this without affecting is, so we can slowly remove the water content by replacing with either ethanol or methanol. So start with the lower concentration, like 10% ethanol, 90% water, so or buffer. So slowly you can increase the concentration of methanol or ethanol and decrease the buffer or water. Finally, you will end up with 100% ethanol or methanol. So that is the stage we called it as complete the dehydration process. During this process, when you go for lower concentration to higher concentration slowly, that will preserve your samples, to hold your samples without affecting anything. So the water content is slowly reduced to the point that the tissue is in 100% solvent and is thus completely, we called it is dehydrate your samples. So next is, like now you dehydrate your samples. So next goal is you wanted to hold your samples to cut for cutting. So how do you hold your samples? It is very, very small pieces of 4 mm or 5 mm, 10 mm size. So how do you hold the samples? It's very tough. Whereas in home, when you cut the samples, vegetables or tomato or anything, you can hold that one with your hand so that you can take a sections, slices. And also no need to worry about the every cut when you do the same size, thickness. Whereas here, every cut you have to have the right, size cutting, whether it is a 5 mm thickness or 10 mm thickness, we have to make sure that. So for that reason, you have to hold your samples. So for light microscopy, we use paraffin. Paraffin is like a candle wax, so we can use that for to hold your samples. So that means if it is a hold your samples, means it has to be, aligned, it has to be penetrated to the tissues, the one which you are going to take as sections. So for reason to facilitate the paraffin wax into the tissues, we can add some chemicals that will be giving an opportunity to penetrate this. So requirements for cutting any material into thin slices. Also, inducing hardness and mechanical support when we, use the paraffin wax. Also accomplished by lowering temperature or infiltration with some material that can be harder. That's also another reasons we use. And also we can, when you do the embedding sectioning during that time, the plasticity, resiliency as opposed to brittles. So when you process your samples, we have to be careful. And this plasticity resilience this one will apply for electron microscope. That statement we will discuss later. So always we are handling the different type of materials, bacteria, fungus, different tissue types. You can use animal tissues, human tissues, cell lines, spores. There are so many different type of materials you use. So for that reasons, we can hold the different type of, mold to pour this. So for that, one of them is tissue blocks. So the tissue blocks is samples handled as blocks may be embedded by placing them in the bottom of bean capsules or gelatin capsules. So this is like, as I mentioned earlier, a small piece of samples you need to hold your samples. So for that, we use a paraffin. So that, we have a different type of mold. One is a flatten, the other one capsule. The tablet, you have a capsule. So you fill the liquid paraffin, and then you add your samples and close it. And at the room temperature, the liquid paraffin become, hardened. So that is one. The next is we have a different type of cell lines we grow in the laboratory. So that is the monolayer of cells may be embedded on the supporter. Um, you can use that, for example, millifer filter or thin plastic. You can use this. Again, the single cells like spores or bacteria, that also we can use the bean capsules, and every time when you centrifuge, that will go down, and then you can, embed it. So this is the one, fixation, dehydration, infiltration, embedding, sectioning, and staining. The polymerization is for different, electron microscope, that steps. So now you have for light microscopy, fixation, dehydration, infiltration, embedding, sectioning, staining. So the fixation, as we detailed now about how do we fix, why do we want to fix samples, and what type of chemical we use, we detailed. So the fixation is very, very important. Most fixation procedures require the immersion of specimens in solutions, right? It is a chemical fixation. So you need to fix the whole material. That means it has to be filled with the fixatives. Also blocks. If tissue is large, if you have a large sample size for 10 mm or 15 mm, 20 mm, something, if you wanted to study like this kind of things, you have to cut the tissue into small pieces. That's the best way to process your samples. Also, already we discussed this monolayers, single cells, like the bacteria or spores, if you wanted to process your samples. Every time, if it is a tissue, just you can take a, tissue using the forceps to transfer from one tube to another tube. Whereas spores or bacteria or fungus or single cells, how do you transfer from one to another tube after you process done? So what the easy way to address this is, you can centrifuge every time after you process. Then you remove the supernatant or whatever chemicals, then you add and then mix it and incubate it and then move forward. So that's the way you can, process your samples with different materials or different tissues. Also, like sometimes what we do is, we have a Petri plates, and we can pour the agar. So that, the media agar, like a 7% or 2.5%. Then, once it is solidified, we take a monolayer, or we take a bacteria or fungus for spores, we drop 1 or 2 drop on the surface of the agar, then it will be absorbed. So then we cut the small pieces of that, and then you can process for the subsequent steps. Another is agar or gelatin. For a single-celled and very small organisms, bacteria, protozoa, fungi, microscopic plants, and animals. Colonies on agar plates may be flooded with fixatives, then pieces of agar cut out for further processing. That's what I explained now. Also, the liquid cultures may be every time you can centrifuge each steps, and then you can, process. So the aldehydes is the formaldehyde, which is available either powder or aqueous solutions. It's a low molecular weight, makes it one of the best penetrating, all of fixatives. So which is commonly used for seeds, spores, plant materials. So this is already we repeated this. So far what we discussed is processing the samples for light microscopy. How do we process the samples? Fixation, dehydration, infiltration, embedding, sectioning, staining. So fixation is formaldehyde or a combination of formaldehyde and glyceraldehyde. You can fix the samples. You can dehydrate the samples with either ethanol or acetone from lower concentration to higher concentration. Then infiltration is to infiltrate your, paraffin into your materials so that we can use that infiltrations. And then embedding. So embedding is just you pour the samples on the capsules or flat bottom mold, and then you add your samples. Always, we will discuss the detail with the processes. So the paraffin is wax. It is like a candle. At a room temperature, it is a wax, solid. When you, heat, then it will be melted. So always we can use the melted paraffin for our experiments. So then only it will penetrate into the tissues. So how do we, the process all of them? So that we'll discuss, next week. Okay. So see you all next week. If you have any questions related to today.